Hi everyone, welcome back to my A-level biology lecture series. In this lesson, we will explore the Krebs cycle. At the end of this lesson, you will be able to outline the Krebs cycle, explaining that oxaloacetate acts as an acceptor of the two carbon fragments from acetyl coenzyme A to form citrate, which is converted back to oxaloacetate in a series of small steps. And you will be able to explain that the reaction in Krebs cycle involves decarboxylation and dehydrogenation and the reduction of the coenzyme NAD and FAD. Let's get started. The Krebs cycle. So the Krebs cycle, also known as the tricarboxylic acid cycle or the citric acid cycle. It is a series of enzyme control reactions that occur in a closed loop. The primary role of Krebs cycle is the release of hydrogen atom, which is very important because it will be used in oxidative phosphorylation to generate ATP. So the diagram shows us how this citric acid cycle or Krebs cycles occur. So generally, Krebs cycle occur in the mitochondrial matrix where acetyl coenzyme A brings or shuttle the acetyl group into the Krebs cycle. Through a series of reactions, hydrogen atoms are harvested. So the hydrogen atoms are very important to be used in the oxidative phosphorylation. So when these hydrogen atoms are removed from the substrates or for the chemical, these hydrogen atoms are transported or transferred to the NAD or to the FAD. So this NAD and FAD, when they receive this hydrogen atom, they themselves are reduced to form reduced NAD and reduced FAD respectively. Let us look at this Krebs cycle in detail. Okay? Krebs cycle occurs at the mitochondrial matrix. It starts with the oxaloacetate, which is a 4-carbon compound. So this oxaloacetate acts as acetyl group acceptor by combining with this acetyl group, 2 carbon, to form a 6-carbon compound, which is known as citrate. In this reaction, coenzyme A acts as a carrier to carry this acetyl group from the link reactions enter into the Krebs cycle, and coenzyme A is released at the end of the first step. Citrate is a 6-carbon compound, and in order to regenerate back oxaloacetate, which is a 4-carbon compound, definitely citrates must undergo a series of reactions involving decarboxylations and dehydrogenation. So after the formation of the citrate, citrates undergoes decarboxylation to form 5-carbon intermediate compound. So in this reaction, CO2 is released. So therefore, the citrate 6-carbon compound is converted into 5-carbon intermediate compound. And this decarboxylation occurs together with dehydrogenation. So in this dehydrogenation process, citrates donate hydrogen atom to NAD. So therefore, in other words, citrate is oxidized to form 5-carbon intermediate compound. At the same time, NAD receives hydrogen atom from citrate, and it itself is reduced to form reduced NAD. 5-carbon intermediate compound must undergo another round of decarboxylation because the whole idea here is to regenerate back oxaloacetate in a series of reactions. So in order to do so, 5-carbon intermediate compound must undergo another round of decarboxylation where the carbon atom is removed as carbon dioxide. And these decarboxylations occurs together with hydrogenation. So therefore, NAD must accept hydrogen atom from this 5-carbon intermediate compound to form reduced NAD. So the first 4-carbon compound, again, will undergo a series of reactions to regenerate back oxaloacetate. So the first 4-carbon intermediate compound is converted to the second 4-carbon intermediate compound, and during this process, ATP is synthesized. So there is a direct transfer of the phosphate group from the first 4-carbon intermediate compound to ADP to form ATP. So this mode of phosphorylation is known as substrate-linked phosphorylation, where ATP is formed during Krebs cycle. The second 4-carbon intermediate compound will undergo 2 rounds of dehydrogenation. The second 4-carbon intermediate compound undergo dehydrogenation to form third 4-carbon intermediate compound. During this dehydrogenation, FAD is involved. FAD receives hydrogen atom from the second 4-carbon compound to form reduced FAD. So therefore, the second 4-carbon compound is oxidized to form third 4-carbon compound. On the other hand, FAD receive hydrogen atom, it itself is reduced to form reduced FAD. In order to generate back the oxaloacetate, this 4-carbon intermediate compound must undergo dehydrogenations again. So these dehydrogenations involve the removal of the hydrogen atom from the third 4-carbon intermediate compound, and this hydrogen atom is transferred to NAD. So NAD receives this hydrogen atom and reduced to form reduced NAD, and oxaloacetate is regenerated. So through this overall reaction, we managed to harvest hydrogen atom from the compound. So in this case, you can see that the NAD and FAD, both of them are hydrogen atom carriers. So they carry the hydrogen atom for the next reaction, which is oxidative phosphorylation. So a quick summary in this case, we start with the formation of the citrates, where the acetyl group from acetyl coenzyme A combine with the oxaloacetate to form citrate. So during this reaction, coenzyme A is released. So therefore, coenzyme A does not involve in the formations of citrate directly. Decarboxylation occurs after that. So citrates undergo decarboxylation, losing one carbon dioxide molecule to form 5-carbon intermediate. And the 5-carbon intermediate undergo a further decarboxylation, losing another CO2 molecule to form 4-carbon intermediate. So in total, 2 CO2 molecules are released per cycle as metabolic waste. Together with the decarboxylation, dehydrogenation occurs as well. So at various stages, hydrogen atoms are removed and accepted by NAD and FAD, forming 3 molecules of reduced NAD and 1 molecule of reduced FAD per Krebs cycle. So this reduced NAD and reduced FAD transfer high energy electron to the electron transport chain for oxidative phosphorylation later. How about the ATP production? So during the conversion of 4-carbon intermediates to oxaloacetate, 1 molecule ATP is generated per Krebs cycle through the process known as substrate-linked phosphorylation, because there is a direct transfer of the phosphate group from the intermediate compound to ADP in order to form ATP. And finally, we have regeneration of oxaloacetate, where this 4-carbon intermediate compound undergoes a series of reactions to rearrange the chemical structure back to oxaloacetate. acetate, which is a 4-carbon compound, and oxaloacetate act as the acetyl group acceptor. So this allowed the cycle or the Krebs cycle to continue when a new acetyl coenzyme A molecule enters into the Krebs cycle. So what are the overall products of Krebs cycle per glucose molecule? As we know that each glucose molecule produces 2 molecules of pyruvate, and therefore, after the link reaction, there are 2 molecules of acetyl coenzyme A. So since one molecule of glucose produces 2 acetyl coenzyme A, so per glucose molecule, the Krebs cycle runs twice. So what are the products formed per glucose molecule? So we know that in one Krebs cycle, 2 CO2 molecules released. So per glucose times 2, we are going to have 4 carbon dioxide molecule. 6 molecules of reduced NAD are formed. 2 molecules of reduced FAD are formed, as well as 2 molecules of ADP are formed through substrate link phosphorylation. This is extended learning, okay, where we look at the Krebs cycle step-by-step breakdown. Again, it is not as simple as we learn in A level. So just for your information, so we can see that it starts with the oxaloacetate to form citrate by accepting acetyl group from coenzyme A. So the citrates will undergo a series of reactions. So you can see that the red color reduced NAD and the red color reduced FAD are formed. So generally, the Krebs cycle, the idea here, the role of the Krebs cycle is to harvest hydrogen atom from the substrate or from the chemical compound here. So with that, we have come to the end of this lesson. Again, I hope you have found these lessons helpful. If you have any question, feel free to leave them in the comments section. See you in the next lesson. Have a great day.